I recording? I should be. Alright, I'll let a few more people come in, and then I'll jump right into biochemistry, followed by physics, and then organic chemistry. Alright, let's just jump right into it. My name's AJ. This is the final part of my series on cumulative MCAT reviews. This is going to be the super long episode. It'll be covering biochemistry, it'll be covering physics, and it'll be covering organic chemistry. Essentially, this is made specifically for the April 9th test takers. So I'm trying to get this out as quickly as possible for them. I've structured this so I'll be covering all the important details, nothing too specific, not the nitty-gritty stuff that you'll be seeing in biochemistry or organic chemistry, especially not in physics. I'll just be going over stuff that you were guaranteed to see on test day, the big picture stuff. So with that being said, let's get comfortable. You guys are going to be here for a long time. Let's hop right into it. We'll start with biochemistry, and we'll start off with proteins. So with proteins, before we, hang on, let me, oh, I'm so sorry, I haven't connected my, my board. All right, there we go, that's much better. So with proteins, before we get started, I think we should start looking at the most important proteins, which are your amino acids. These are guaranteed to be on test day. If there's any one thing I want you guys to learn from biochemistry, it would be your amino acids. So with that being said, we're going to learn the structure, we're going to learn the backbones, we're going to draw these out together. I want you guys, watching, I want you guys to draw this out with me. All right? So I'll start off with the basic backbone. The skeleton of an amino acid is something like, I wish I had a bit more space, but I'll just draw it here. I'll start off with the N-terminus. And then here is the C-terminus. Now, whenever you're reading, writing, looking at an amino acid, just know that you're going from N-terminus to C-terminus. Here, let me make this look a slightly more legible. There we go. And then this right here is going to be an R group. This right here, that's the one that changes in every single amino acid, and that's the one that gives the character its defining traits. Now let's move on to our non-polar amino acids. These amino acids, I'll go over the acronym for it later. I'll just write them out. Let's start off with the easiest one, glycine. I'll write the amino acid skeleton, just AA. So this one is called glycine. Actually, no, I'll just do the 3-letter and then the 1-letter. Glycine, followed by G. I'll write all of them out first, and then I'll write them out. Actually, no, I'll do it one by one. Let's do alanine. This is just going to be a CH group. No, I probably should just do it all by one. That would be more efficient. Let's do valine. Isoleucine, or I'll do leucine first. Leucine. Isoleucine. Methionine. And I wish I had a little bit more space for proline. I'll try and squeeze it in here. Never mind, I don't have the space. We'll try and draw it somewhere here. I'll draw it like this. All right, let's start labeling. So this is glycine. I said this was alanine. This was valine. This is leucine. Isoleucine. Methionine. And this is proline. Now, let's talk about proline really quickly for a second. Proline is a very important amino acid because this is the one that you'll be seeing in your secondary protein structures, your alpha sheets, or sorry, your alpha helices and your beta sheets. This is what you'll be seeing. This is what causes the kinks in the alpha helices and beta sheets. It's what causes like the alpha helices to like be helices. It causes the beta sheets to like start pleating like this. It prevents it from forming a straight like line, basically. Know that for the MCAT, they like testing that. If I can draw like 1,000 stars around that, I definitely would. But I'll just draw one just so you guys can get that through your head. With that being said, let's move on to your nonpolar. The acronym. It's kind of vulgar. I'm sorry if you don't like it, but the one I use for nonpolar molecules is limp-vag. It basically covers everything that you need. I don't think there's, I don't know any acronym that works like limp-vag. If you have one that you like that's less vulgar, go for it. Tell me in the chat actually if you have one, because I'd like to use anything other than limp-vag. With that being said, let's move on to nonpolar aromatic. Nonpolar aromatic. These are the aromatic ones. These are easy to memorize. So let's start off with phenylalanine. Tyrosine. Oops. I hope you guys are drawing along with me, by the way. These are very important. And tryptophan. So this is phenylalanine. That there's an F here because it's phenylalanine. Tyrosine with the Y. And this is tryptophan with the W. The way I'd recommend from memorizing these. Phenylalanine tyrosine, they're the exact same, but tyrosine has an extra OH, giving it a slightly polar character, but overall it's a nonpolar. That's why it's under the nonpolar aromatics. Just some fun trivia there, I guess. Don't want to confuse you guys with anything else. Let's move on to polar amino acids. I think these are the hardest to memorize because there's no easy. They don't have easy structures. They're not like there's no like lymph-veg. There's nothing like that you can use to memorize them. So just like I don't know, like memorize this like raw or like use rote memorization. Let's start off with serine. Threonine. Cysteine. Glutamine. And what's the last one I'm missing, guys? Asparagine. Alright, so this is serine, threonine, cysteine. What did I say this one was? Glutamine. And this is asparagine. These are, I guess asparagine isn't that bad because there's an N in there. Make sure you memorize that there's an N so you can get that. Glutamine's a complete wild card. I don't know why there's a Q there. Scientists are weird. So just memorize that glutamine is Q. I like to talk about cysteine for a second because this one is also very important. Why is this important? Because in tertiary structures, cysteine is the molecule that is used to form the disulfide bridges. So one molecule of cysteine and another molecule of cysteine, so like this is attached onto another amino acid, they will connect together, removing the hydrogens to form water. They will connect together and form a disulfide bond. I'll get more into that in later in the lecture, but just know that this is a very important molecule because of the disulfide bond character. All right, now negatively charged amino acids. There's two of them. One is aspartic acid. I'll write them on top of each other. So and glutamic acid. Oh, whoops. Man, I shouldn't have written them on top of each other. I'll draw it a little bit further out so it doesn't like mix up. How many are there? 1, 2, 3. Yeah, I was right. 1, 2, 3. 1, 2. I'm so sorry, guys. I got this confused. Okay. 1, 2, 3. Yeah, maybe I should have just drawn out the entire thing. It would have been easier on my head. But I don't want to crowd out the board. So this one, the shorter one, is aspartic acid, ASP or D. And this one is glutamic acid, GLU or E. Way to memorize these ones, I would say, is that the shorter one comes before the longer one. So D comes before E. So just think about if you're negatively charged, you have D and E. And the shorter one comes before the longer one. Now the negatively charged ones, there's 3 of them. Or the positively charged ones, sorry, there's 3 of them. These are, I like to point out, by the way, that these are acidic. And these will be basic. Yeah, I know I underlined that a lot. I really want you guys to understand these amino acids. So with that being said, what were we going into? We're going to the positively charged ones. So let's talk about, I'll start with 1, 2, 3, 4, and then followed by the NH2. This is lysine. Now let's move on to arginine. I'll start off with the NH. That'll be easier. You guys can draw this however you want, by the way. One, two, three. Not enough space, all right. I'm meshing into the protein structures. I wanna try to make this a bit shorter. All right, there we go. So this one is lysine, the K. This one is arginine with an R, and this one is histidine with an H. Histidine's pretty straightforward. Arginine, remember there's an R in there. Lysine, it's like glutamine, it's a wild card. It's just a K. That being said, I don't have anything interesting to say about these. I'll go back to the negatively charged ones for a second. Since they're acidic, they have deprotonated forms called aspartate and glutamate. They don't have the hydrogens attached; they'll just have a negative charge. But that being said, I think that's all you need to know for amino acids. Know their structure, know the 3-letter forms or the 1-letter forms. I'll get into properties, some more properties later on. It'll make sense once we get into like some more biochem. Next up is protein structure. I know we have a little bit of like an overlap here. I think this is enough space though. So we have right here the primary, secondary, tertiary, quaternary. I'll keep it there, but just note that this is not that important. I'll star the ones that are. I think tertiary is very important just because there's so many bonds that are going on there. So primary protein structure, it gives you like the backbone. I don't want to say backbone. It gives you like the blueprint of the molecule. It's basically what forms like its code, its most basic properties. And they're secured together, they're attached to each other via peptide bonds. And I'd just like to make a quick statement. Peptide bonds is when the C terminus, let's say if this is an amino acid 1, the C terminus of 1, and this N terminus of another, amino acid 2. They go through a dehydration synthesis reaction and they form a bond. Now secondary, this is the alpha helices, your beta sheets. You'll be familiar with this in biochemistry. Like I said before, it's associated with proline. That's what causes all the kinks, the folding, and all that. The bond that's associated with these is hydrogen bonding between the backbones. So like the amino carboxyl backbones of side chains. I'll write R groups. Tertiary bonding. There's tertiary bonding. That's the 3D shape. I'll get to like a way to memorize it in a second. But the 3D shape, it's associated with hydrogen bonding as well. It's hydrogen bonding between adjacent R groups. So the backbones in the secondary bonding, they'll be from like here to here. That's forming the structure right here. You'll see it in more detailed diagrams, you probably have. But with their tertiary structure for folding and all that, it's adjacent side chains. Now, as one of my students that I was tutoring one-on-one, he told me that the way he memorized amino acids or protein structure in a way that made it digestible and for him to understand what it is that he thought of the primary protein structure as the words or the letters in a word. Like they're the most like basic units that you're using it to create meaning. Secondary protein structure is when you have some meaning, you have like an idea of what it's going to lead into. These are the words that are present in a sentence, which would be your tertiary. Tertiary, a sentence, it has full meaning, it's a full unit, it can tell you exactly what you want. And quaternary, I guess, would be like a paragraph. Quaternary is like multiple tertiary bonds connecting together to create like a large protein. I wouldn't worry too much about that. Just know that that's what it is if they ever do ask you about this. That being said, we already did a lot. Let's move on to enzymes. Now, enzymes, you guys know what enzymes are. Lowers the activation energy, stabilizes the transition complex. What else? That basically covers like the basic stuff, like the high school biology stuff. Let's move into like the more advanced stuff. Let's talk about the different types of enzymes. Do you guys know what Lil Hot is? No? Okay, so I'll write it out. So Lil Hot is your lyase, isomerase. Oops. Ligase, hydrolase, oxidoreductase, and transferase. So lyase, it basically like lyses bonds, cuts bonds in two. Isomerase, it just creates an isomer of your original substrate. Ligase, it fuses things together. Hydrolase, it causes a hydrolysis reaction. It lyses just like lyase, but it specifically works for hydrolysis. Oxidoreductase, it works with redox reactions. And transferase transfers functional groups. I know I went by that really fast. Does anyone have any questions? Okay, cool. So I'd like to point out that the hydrolases, they have very different names. Because most of them, they'll just be like X lyase, blank X isomerase. But like with hydrolase, these are your proteases, your nucleases, lipases, stuff like that. They're named differently. Now let's move on to, let's talk about what's going on with active sites, allosteric sites. I wish I put in an image, but I'll just draw like an enzyme right here. If this, and then. That's a bit better. So this is an enzyme. We'll say this is an enzyme. This is your substrate. And let's say that it's binding to the active site here. Active site is like the direct binding, it binds right there, and then it creates, it activates the enzyme to form catalysis. If it was attaching on to the allosteric site, however, it would change how the enzyme functions, or not functions, it changes its state. It makes it look different. And then it would open up to be bound onto an active site. So now that we know what allosteric and active sites are, let's move forward towards how competition affects it. Competitive inhibition, it's the most straightforward. It binds directly onto the active site. So, well, let's say like this is a competitor. If it binds here, it prevents the substrate from binding, preventing any more of the substrate from being catalyzed. So when you have an active site inhibitor, a competitive inhibitor, you will be increasing the Km. Km is the concentration constant. The Vmax, however, since you're not you're just substituting the substrate, it won't be changing. There will be no change. Now let's move on to non-competitive. Non-competitive, it binds to the allosteric site. Whoa, what? Oh, it's so weird. What happened? I'm sorry, guys. We're having some technical difficulties. I guess I'll just refresh the page. That's much better. Yeah, I'm sorry, guys. That's never happened to me before. So, where was I? We're binding to the allosteric site for non-competitive inhibitors. So when you bind to an allosteric site, you won't be changing the Km, but you will be decreasing the Vmax. So moving on to uncompetitive, this is when you're binding not to an allosteric site or an active site, but you're binding to the ES complex as a whole. So even if there's a substrate attached, so let's say a substrate does bind, all right? But you have an inhibitor coming in. I'll do it as like, I'll say it's this. An inhibitor comes in, it binds to the complex anyways. Not necessarily to the, it could bind to the allosteric site, but so it binds there. Both are present, and it changes it structurally and functionally. So when you bind to the ES complex, the enzyme substrate complex, you'll be decreasing both Km and Vmax. Now the last kind is mixed. Mixed is like sort of like it's a mixture. It could be a little bit of this. Like some enzymes could be inhibited competitively, others can be inhibited allosterically. So like it would be present primarily on the allosteric site, however. And it would cause an increase or decrease on Km, just depends on the concentrations. But it will always decrease Vmax. So moving forward with the Michaelis-Menten, this is Michaelis-Menten, this is Leinert-Weaver-Burke. That's a bit big, I don't want to waste a lot of space. Michaelis-Menten, So I'm going to write the equation too. So Michaelis-Menten, the only thing that you really need to know from this chart would really be that, oh, oops, I almost erased everything. Right here, at the halfway point, that's at half Vmax, that's where you will find your Km. So this is rate, this is volume, velocity. Note that after you reach your half Vmax, you go from a linear graph to it's more sigmoidal, or not sigmoidal. It's more logarithmic. I'll get to it's pressure based. So move from high pressure to low pressure. Yep. And this right here is the structure of a G protein coupled receptor. I think we all are pretty familiar with that, but just in case we're not, I'll walk you through it. So a signaling molecule signal molecule binds to a receptor protein. That receptor protein then sends a signal to activate the G protein coupled receptor. And then that G protein coupled receptor sends a secondary messenger moving forward. This is actually a pretty bad diagram, I won't lie. But it would send a secondary messenger moving forward to another molecule, and then that would create a cascade of response. Usually the one that you have to know is CAM. If not, there are some that they'll give you in the question stem or in the passage itself. But CAMP is usually your secondary messenger that will most likely be tested. Ooh, let's move on to carbohydrates. Carbohydrates. This is it's a smaller unit, but it's still very, very important. So let's talk about aldoses and ketoses. How do you guys want to do the breaks this time? Do you want me to just finish all of biochemistry and then we do a break? Yeah, I'll finish all of biochemistry, no worries. I don't think we have... we have a good amount to go. But that's fine. It's fine. We'll be okay. So, carbohydrates. Carbohydrates. Where do I want to start? Let's start off with just structures. You're seeing that this is a ketohexose, aldohexose. Aldoses have aldehydes present in them. Ketoses have ketones. Pretty straightforward. The hexose, it's obviously a 6 carbon ring. If you have a 5 carbon ring, you'll be a pentose. 4 carbon ring, tetrose. You'll never see that really. 3 carbon ring, triose. One thing that I also want to talk about would be the anomeric carbons and the alpha and beta structures of them. So an alpha structure of a sugar or carbohydrate, alpha anomers will have the this OH right here present. It'll be trans to the free CH2O2 group. It'll be trans to this one. But it's if it's beta, it'll be actually cis. Yeah, I'll explain what an anomeric carbon is. An anomeric carbon is the new chiral center that is formed. When you create a ring, it's the new chiral center. So, moving on from that, let's move to some monosaccharides. So we see fructose right here. I'd like to also point out that all natural carbohydrates will be in the D form. You'll rarely see L form. It's probably going to be in a lab situation. They probably won't test you on that either, but just memorize. Just make sure you know that it'll all be present in the D form. For fructose, how would I want you to memorize this? Note that there is a double bond. There's a carbonyl group here for fructose. So note that there's a carbonyl group. For glucose, you see that there are 3. I drew that in a bit thick. There are 3 hydroxyl groups present on the right side. This is D-glucose, so it's always going to be present on the right side. If it's L-glucose, it'll be on the left side, but we'll always be focusing D-glucose. So on the left side, there's 3 of them. One of them is separated by just hydrogen. You'll never see, obviously, you'll never see all 4 on one side due to steric issues. Galactose, you have 1 at the top and 1 at the bottom. And mannose, this is a bit rare. You'll see 2 at the bottom. That's how I like to memorize them. If you guys have any other better ones, feel free to do so. I think I'll just draw like that. 3 OH. OH on top and bottom. 2 OH on bottom. Can't really think of anything else for that. Any other questions about monosaccharides? We can move on to some disaccharides and polysaccharides. Sucrose is basically just glucose. 1, 2. Sorry, I messed that up. Glucose. 1 alpha 2 fructose. And I'll do lactose as well. That's galactose. Beta 1 for glucose. L and let's do maltose, why not? Yeah, I know I messed up the first one. Sorry guys. This is just something you have to memorize. I don't have any like cool ways to memorize it. But yeah, that's pretty much it. We're moving on to another shorter unit in membrane transport. So let's start off with a refresher on osmotic pressure. Van Hoff factor, that's going to be like the number of molecules present, so that when you dissociate it, so like glucose, it won't have a larger like I number. It's always going to be 1 because it's not going to dissociate into anything. Ionic compounds will, however, so just take note of that. M, molarity, R is a constant, T, temperature. Note that if this is a I'm not sure how you say inverse as like a symbol, but it's not proportional to osmolarity. Now, passive transport, this will be, what did I just say? No, osmosis. Yeah, so this would be your passive diffusion. Also known as simple. I'll write it as simple. Now, simple diffusion, we know that it doesn't need a transporter. It's basically just go down the concentration gradient high to low. Facilitated diffusion. In this one, you're using a membrane transporter that'll help you move it across cell membranes, and osmosis. In every situation to the MCAT, know that osmosis is related towards water. Okay, yeah. I'm always so paranoid about that stuff. Sorry, I'm just trying to make sure that I wasn't muted. Active transport, it's a bit different. You're using energy. That's what the definition of active transport is. You're using energy, and it's in the form of ATP, obviously, and you want to move against the concentration gradient. So, that being said, let's talk about uniporters, symporters, antiporters. Uniporter, it moves it from one direction only. Symporters, it moves two molecules down into the cell or out of the cell, same direction. One will be going towards its concentration gradient, and the other one will be going against. They're both working together to get into the cell. Antiporters is the complete opposite. They're both going against the concentration gradients. Or one's going with its concentration gradient, sorry, that would be A, and one's going out against its concentration, that would be B, and it's just trying to be moved out of the cell. That said, I have some notes about endocytosis, exocytosis, pinocytosis, phagocytosis. I feel like those are pretty straightforward. I don't think we need to talk about that. Just know that phagocytosis is like the cell eating, pinocytosis is like the cell drinking. DNA. This is a very important one, guys. DNA and RNA, these are very important. Sorry, I think I got something in my eyes. Let's start off with obviously the model. So this is a nucleotide. Nucleotide has a nitrogen space, it has a carbon sugar, it has a phosphate group. A nucleoside, however, will not have a phosphate group. Side means it doesn't have the third side. It doesn't have the phosphate group. So nucleosides, they're not important. Let's move on. I was thinking about like where ATP is, and I'd like to point out that these bonds are very, very high energy bonds. These are the ones that you'll be breaking off to make stuff like ADP, or reforming to form ATP. Very, very high energy bonds. They're also hydrogen bonds. So I'd just like to make sure you guys know that. You guys know the Watson-Crick model, right? DNA goes from 5' to 3'. It's anti-parallel. Double helix. You know your purines and pyrimidines? That was from the biology lecture. If you guys didn't have a chance to watch that, I'll write it down anyways. Purines are A and G. Let me make some more room for all this. Pyrimidines. C, T, and U. I'll write this so it makes more sense. I made the mistake in my previous lecture, C, T, and U. I'd like to point out that the pyrimidines, there's 3 of them forming a triangle like a pyramid. So 3 pyrimidines, 2 purines. Make sure you have that figured out. They'll ask you a question about purines for sure. Since I had to cover it in biology and biochem, you're probably going to see it. I'd like to point out that these bond with triple bonds, these bond with double bonds. I'll do that in blue. And this is pretty obvious, like we've been through this so many times. This is only present in uracil, is only present in RNA, thymine is present in DNA. One thing I'd like to talk about is heterochromatin and euchromatin. I don't have a fun way to memorize these, but heterochromatin, it's dense and it's silent. It's DNA, it's tightly wound up. It's present in your histones. Euchromatin, on the other hand, it's loose and active. Active and silent, I'm talking about in the presence of transcription and translation. Mostly transcription. You guys know what telomeres are, you guys know what centromeres are. Let's move on to some of this stuff. I'm not going to read all this out, you guys can read for yourselves. Basically, the step and where it is in the prokaryotic and eukaryotic cells, it makes sense. Basically, DNA polymerase 3 and 1. Just know that that's pretty much only for prokaryotes. In eukaryotes, we have it named like alpha and sigma. Here's one I got wrong. I hate to admit it, but I got this wrong twice on practice problem. Prokaryotic cells do not have telomeres, guys. So, yeah. That being said, let's move on to this diagram here. I'd like to talk about leading and lagging strands. Leading strands and lagging strands are synthesized by DNA polymerase, or mostly the leading strand. It's still synthesized by DNA polymerase. It goes from 3' to 5' when it's reading the template, but it creates a new strand from 5' to 3'. Now, this is only present in the leading strand. Okazaki fragments are present in the lagging strand, and they're basically fragments of DNA that are made in like chunks. They're made with primase. They're creating like little pieces and they stop, little pieces and they stop. That's the lagging strand. And then they're later filled in by DNA ligase. I'd like to point out that DNA is semi-conservative, meaning that the original strand is technically conserved, but it's used in a way that it can create a new daughter strand. You guys can see that it says semi-conservative. I'm trying to think. Do I want to introduce Snowdrop now or after RNA? I'll introduce Snowdrop now. I have the room. So you guys are thinking, like, what is he talking about Snow for? So Snowdrop is an acronym that I use to memorize 3 very important tests or techniques that they use to determine like proteins, DNA, or RNA. So let's think about it in a way like Snowdrop. No. There is a thing for the O's. It's just used for the sake of the acronym. These tests detect the presence of DNA, RNA, or protein. So with Snow, this will be a southern blot. That's used to detect DNA. Now this is a northern blot. Used to detect RNA. And then western blots, sorry. Use that to detect proteins. Now I know probably all of us have done western blots in our biology labs. In my research lab, I was able to do northern and southern blots, but I don't think everyone else has. So if these are difficult for you, I highly recommend just knowing Snowdrop. That's all you really need to know. With that being said, let's wrap up DNA and let's move on to RNA. Now, we all know central dogma, right? We know the central dogma of biology. DNA, RNA, proteins. With the genes, though, what do I want to talk about? I'll start off with, it's basically just codons. Start codon is AUG. It's always going to be methionine. Alright, start a bit better. That looks like strat. And then now stop codons. These are very important. I remember when I was teaching my little sister like biology, because she's younger than me, and she's always like just very confused on the stop codons. And one thing that we came up with together, you guys probably heard of it too, I think it's pretty common, is you are away, you go away, and you are gone. Now these are your 3 stop codons. I should probably write those. You are away, you go away, you are gone. Cool. Now, with the types of RNA, I was thinking about adding mutation stuff here too, but we covered that in the biology lecture. That honestly covers it like super in depth. I think you should just watch that instead. RNA, so messenger RNA. Now this is the messenger RNA. This is the kind that transfers or carries the message of the DNA that's present in the nucleus and transfers it to the ribosome for translation. Yeah, there's no way I can write all that. Just goes transcription to translation. I'll just say it's messenger. Or nucleus to ribosome for translation. tRNA, this is transfer RNA. Now this is the one that brings in amino acids in translation. For translation, it allows the codon to recognize what is present on the mRNA to create an anticodon. So I'll just say brings amino acids for anticodon. Ribosomal RNA, honestly, just know that it exists. It's basically just the structural unit of the ribosome. That being said, let's move on to transcription and translation. Transcription, this is a kind of a, I didn't want to get too complex of a diagram, but there's an initiation step, an elongation, and a termination step. Basically, the helicase and topoisomerase will unwind the DNA double helix. There's just something that's called RNA polymerase, this one right here. It binds to a promoter sequence, which is called the Tata box. And then it's called the Tata box because there's lots of AT bonds. And it synthesizes RNA from the DNA template. And then that's basically it. I'm trying to think of some post-translational stuff that goes on, or post-transcriptional stuff that goes on. They usually splice it. It's done by spliceosomes. They'll create introns and exons. Exons are relevant DNA. These are usually ones that will be coding for something. Introns are the ones that are spliced out. They add a poly A tail to the 3' end just to know that the transcription is done and can be sent out to translation. That being said, let's just talk about translation. 3 stages: initiation, elongation, termination. I'm trying to think. It's the codon stuff. It starts with the start codon, keeps moving in groups of 3 to create anticodons that'll be able to code, and then it stops eventually when you reach a stop codon. Each of these codons and anticodons, they code for an amino acid. The post-translational modifications I was thinking about is that they're folded by chaperone proteins, and they usually are playing a role in quaternary structure. That's all I think I have for RNA. That's the hardest stuff, I believe. So I think just kidding. Just kidding. We just got to the actual hard stuff. Metabolism and glycolysis. Good news, MCAT does not require you to know this. Or they don't require you to know all the steps. I know like you guys are looking at like this right here and it's like, oh god, that's so complicated. The TCA cycle looks it's super frustrating to learn all that, but you don't have to know it. You just have to know the important stuff about it. I would say know that the glycolysis needs 2 ATP to start. It forms 4 ATP. So the net yield is actually just 2 ATP. Know that this is anaerobic. And there's some important enzymes. I think glucokinase is the only one that's important. It's just responsive to insulin. That's what starts everything. Hexokinase is what traps the glucose. PFK1 and 2. PFK, I'll write this one. PFK1 is a rate limiting step. That's essentially it. We can now move on to the citric acid cycle. If this occurs in the mitochondrial matrix, this occurs in the cytosol, by the way. This occurs in the matrix, mitochondrial matrix. Its main purpose is to generate electron carriers for the oxidative phosphorylation and the electron transfer chain. So it mainly it's NADH and FADH2. I'll throw GTP in there as well. That's the most important stuff. You don't really need to memorize any of this, like I said. I think just knowing a few of the products. I think malate, fumarate, succinate. If you know those, you know that they're part of the TCA cycle. It's also pretty straightforward. You never see malate anywhere else in biology or biochemistry. Same with fumarate, succinate. It's just for citric acid cycle. And now let's move on to the electron transport chain and oxidative phosphorylation. So pretty straightforward. It just transfers electron carriers from one complex to another. It basically just goes down a chain. One thing that the MCAT would love for you guys to know is this right here. Oxygen is the final electron acceptor. Not carrier, electron acceptor. This is what it accepts, and it moves forward to the oxidative phosphorylation part, which is basically just ATP synthase. It provides the energy in order for ADP to bind to its last phosphate, creates ATP, and it moves forward like that. I'd like to highlight that it generates 36, but in actuality, it's about 30 ATP from oxidative phosphorylation. I'm trying to think, because I don't know if I really want to. Yeah, I'd say like the rest of like the AGP, ATP formation that occurs in the electron transport chain and the citric acid cycle aren't really irrelevant. They're used during those processes. This is your final yield from oxidative phosphorylation. I like to point that out. Compared to the 2 ATP from anaerobic, there's a reason why aerobic respiration is highly preferred. I'll get into like the lactate stuff later. Glycogenesis and glycogenolysis. Tongue twister. I think with glycogenesis, I'll just talk about glycogen synthase. Now it's a synthase, so you know that it's creating something. It is creating alpha 1 for glycosidic bonds. And then the branching enzyme this creates alpha 1,6 glycosidic bonds. And then glycogenolysis, it's basically opposite of glycogenesis. Glycogen phosphorylase. Removes your alpha 1,4 bonds. And you won't believe what the debranching enzyme does. Removes alpha 1,6 bonds. I know I went through these two really fast. There's nothing important that you really need to know about these pathways or any of their products or reactions. Just know what those enzymes do. I guess if I had to say anything, that glycogenesis builds up glycogen. So this one, I'll do it in red. Purpose, increase glycogen. That's why you have synthases. This one breaks down glycogen. Yeah, if anything, that's basically what these pathways are for. Let's move on. I have two more for you guys. Gluconeogenesis and pentose phosphate pathway. Gluconeogenesis is basically reverse glycolysis. There are 3 irreversible steps. So I think it's, I know I'm just going to write it as pyruvate carboxylase and PEP carboxykinase. There's no way I actually fit that there. These pyruvate carboxylase and PEP carboxykinase, they bypass pyruvate kinase, so they and the second one is fructose 1,6 biphosphatase that bypasses PFK1. And then the third and final one is when G6P I write faster than I think. Bypasses hexokinase, or if you're in the liver, glucokinase. So these are your irreversible steps. Now, I gave a lot of space for the pentose phosphate pathway, but I don't think there's a lot we can talk about. Just know that it occurs in the cytoplasm. It uses peroxisomes. That looks like an S. Uses peroxisomes. The rate limiting enzyme is G6P dehydrogenase. And the purpose of it will be to generate NADPH. All right, cool guys. We just finished with biochemistry. I think we're gonna take a break right now. We just went through an entire subject. We'll be back in about 4 minutes. I'll say 4 minutes. And then we'll get right back into physics. See you guys then. Alright guys, I'm back. I think we took a bit longer than 5 minutes, that's okay. But yeah, we're moving on to physics now. Now I'll just be straightforward with you guys. I've been dreading this topic. It's definitely my least favorite subject on the MCAT. Even more than cars. I absolutely despise physics, but we have to know it. We have to learn it. So let's do our best. Let's do our best. Where do we start? I didn't bother doing anything for kinematics or Newton's laws. I feel like that's very almost elementary level of physics. If anybody has a question, I'll wait right here. We can talk about it if you guys want. If not, I'll be moving straight forward into thermodynamics. No questions. All right, cool. So with that being said, let's move into thermodynamics. Let's go. I want to talk with. I'm so sorry, guys. It's pretty late at night. It's like 11 here. But yeah, don't worry. I'm not falling asleep on you guys. We'll get this done. Expansion. Thermodynamic expansion. This is the equation for linear expansion. Now, linear expansion is when the length of a solid is increased when it's heated. It's pretty, like it always, or I wouldn't say always, but it usually is. An easy way to think about it is that when temperature increases, the length of the solid is increased by a lot. A-L-O-T, a lot. These are both constants that they'll give you. Or no, this is the length, but like the alpha is like a constant that they'll give you. Change in temperature. Volumetric expansion is basically the same. It's just delta V equals beta V delta T. That is volumetric expansion. Similar concept. Just give me 1 second. Testing. Okay, cool. Yeah, I am recording. I'm so paranoid, man, because like if I found out I was recording this for like an hour and I didn't have my mic on, like I don't know what I would do. It's already like I don't know. It's just something. It's a little fear in the back of my brain. That being said, back to thermal expansions. I think I covered everything I want to talk about. It's basically just an equation. That's most of physics, to be honest. A lot of it is you can just memorize the equation. You can probably just like fight your way through a passage. Let's move forward with heat transfer. This is your conduction, convection, and radiation. So conduction is the transfer of energy directly. So this is like with your molecules colliding. So like, I'd rather draw than write it out. So it's just two molecules colliding, creates whatever the collision is. That's going to be heat. Convection, it's a bit different though. Convection is fluids. Whether it's liquid or gas, it's fluids. So you're trying to go, like, whether it's flowing this way or it's flowing like both in the same direction of two different things. It's usually separated by some sort of membrane, but the flow between each other, it transfers heat. So from here, it goes here. And I guess a little bit of vice versa. You heat one, you cool the other, right? And radiation, transfer of energy by electromagnetic waves. I don't have a cool way to draw that out. It's just EM waves. Sorry guys, I know you love my drawings. Just kidding. So with that being said, we can move forward to specific heat. I'll move a little bit to the left so I can get some room. How much room do I have here? Not a lot. All right. Q equals MCAT. That we talked a little bit about that in the general chemistry section. But basically, we already talked about it. Just know that when if Q is greater than 0, heat is gained. Q is less than 0, heat is lost. I want to talk a little bit about... I don't even need to use that space. Okay, so let's talk about adiabatic. Isothermal. And isovolumetric. These are, you guys also probably remember this from the Gen Chem lecture. I just want to bring it up once more. This is basically, I'll write the equation out too. This is what we're working with here. Oops. Q minus W? It is minus W, yeah. So in adiabatic process, that's when the pressure is held constant. Q would equal 0. I'll write this in a different color. So there's, I've been using a lot of red, I'll use blue. Q equals 0, so that means delta U equals negative W. Based off of this. Crap, I wrote that for isovolumetric. Sorry, guys. Q equals 0. I'll write for this one W, or this for isothermal, it's delta U equals 0. This is W equals 0. So like I said, when Q equals 0, delta U equals negative W. My thing is blocking right here. Sorry, guys. I'll draw that just a little. Yeah, it's more readable. When delta U equals 0, it just means that Q equals W. Just move that over, right? And then for isovolumetrics, delta Q or delta U equals Q. So when W equals 0, delta equals Q. Basic math. That being said, I'm trying to think if there's anything else I want to talk about. Because this right here is also the first law of thermodynamics. Second law of thermodynamics is basically just saying that the entropy is always increasing. Third law is when the entropy of a perfect crystal is 0. That's basically it. Not really much else I can say about thermodynamics. Let's move forward to fluids. I think fluids is arguably the most important concept in physics that you need to learn. Oh wow, I don't have much here. Why am I glitching? What is going on? Let me... This is so weird. Why am I glitching like this? Alright guys, I think I'm gonna have to stop stream for 1 second. I will be right back. I think I'm just having a glitch on my computer. Just give me a minute, I'll be right back. I'll try and stop streaming. If I can find my... I'm just gonna have to restart. I'm sorry guys, this is weird.